So let's start with genetics. And what is for you the relevance of genetic testing in the overall management of the patients with CMT disease?
So genetic testing is the absolute basis of the diagnosis. So to get a diagnosis, you need that to get into a clinical trial or to access therapies for the future. So it's the absolutely critical point, and to make decisions about, the pregnancies, etc. So I think that's absolutely critical. So it's the first step on the translational circle, going from the patient right around to actually having treatments. So I think it's incredibly important.
Yeah, of course. And are there modifier genes that can influence the severity of the progression of the disease?
So I think that's really interesting. So traditionally, I would say we're now at the end of the first era of genetics. We know the vast majority of genes that are single genes. So in our series, we have about 77% hit rate, meaning that patients that have one gene causing the disease, we know it. But what we realize now is how much we don't know. So we've heard this morning about unusual variations that we'll only pick up by long read sequencing, and maybe even other ones that are hidden. And then the next era of genetics is modifier genes. Why is the phenotype of the same gene different in different patients within a family, and perhaps even more between families? And then modifiers of the genes that might predict our response to therapy. So if you give a gene therapy, why will it work differently in different people? Modifier genes to the potential therapies, and modifier genes to the side effects of therapies. It may be that we realize as we treat patients that the genes that they have for other things, for instance, the immune system. So I think we've only begun the genetics. So it's a new era of it to do with modifying across the spectrum of treatments.
So in this line, in what ways do you think that these genetic insights impact the patient's everyday life?
So I think it's been a huge impact. So I think when I look back to 1991, when the first gene was found, and then the trickle of genes, we were very confident when we saw a patient. This is the cause in you. This is what will happen. And now I think we're much more nuanced in giving advice, because we know the gene is only the beginning. Sometimes we're not 100% sure that gene is causing the disease, and a major problem for patients is being told is a variant of unknown significance, because to get into a trial or a treatment, it needs to be definitive. So we need to be better at doing that. And to me, the major answer is more families with segregation. That means the world getting together to get data in a common way, searchable, and being able to use that to do it. So I think it's very important.
Yeah. And moving on potential therapies, what are the biggest challenge in translating these genetic discoveries into effective treatments?
Okay, well, there are 5 major challenges. The first one is preclinical validation remains a challenge. We heard today people saying you didn't do enough models. You didn't do enough. You've got to get the models properly. And we're lacking in CMT guidance as to what is the optimal. And we've said this to our basic science colleagues a lot. Number 2 is developing therapies that are safe. CMT generally is a lifelong condition. The safety bar is huge. Number 3, and the biggest, is delivery. How we get it to where we want it, that is the peripheral nerve, be it the Schwann cells, axons, or motor nerves, safely at doses that work, systemically rather than intrathecally and without liver toxicity. And fourthly is developing the outcome measures that we can do clinical trials. And then absolutely huge problem is global equity to treatments in due course.
Yeah. So finally, let's look towards the future. How do you envision the diagnosis and the treatment landscape for CMT evolving over the next 10 years?
So I think the involvement over the next few years, the initial involvement will be trials. The trials are already started. We have trials ongoing. They will exponentially increase in the next 2 to 3 years, particularly with the disease-agnostic treatments based on RNA, DNA therapies. They will happen predominantly in adults for the common forms of CMT and eventually children. I think within 5 to 10 years, I hope we'll be getting this earlier. So treating people with very early markers of disease in early childhood with effective therapies to prevent the expression of the disease. And that's where I see it. Do I think we'll be there fully in 10 years' time? No, but I think we'll get there. But humans are humans. There'll always be people that aren't tested and turn up late and in different countries. So we have to keep, this is the optimal, but we also will always have to treat patients who've already established disease. And what we shouldn't forget is, have we got any genetic therapies that could treat disease as people have it rather than prevent it? So could we have a therapy for axonal transport that would treat even if people have established disease? So I think there'll be both pathway therapies for lots of forms of CMT and very personalized individual therapies developed in adults first and then children. And I have no doubt that there will be therapies within 10 years, you'd never say for definite, and multiple trials within 5 years.
Yeah. So thank you so much, Mary.
Thank you very much.